Good afternoon, everyone. This is Wanda Fink. I'm the Director of Operations for the UMB ICTR. Thank you for joining us today in our Enrichment Seminar Series. Today's presentation will be about our relatively new ICTR core of services, the Drug Discovery and Development Core. But before we get started, I just wanted to go over some Zoom housekeeping rules. If you could, please make sure that your microphones are muted. Also, for questions, we are planning to reserve 10 to 15 minutes at the end of today's presentation for our QA session. Please post your questions through the chat feature found at the bottom of your screen. Today's presentation will be hosted on the ICTR website, and that link is going to be posted in the chat feature, as well as a link to a satisfaction survey. Please take a few moments today and let us know how we can improve our presentations and what additional topics you are interested in. A QR code to that survey will also be available on the final slide of today's presentation. So today, our guest speakers are Dr. Paul Shapiro. Dr. Shapiro is a professor at the Department of Pharmaceutical Sciences here at the University of Maryland School of Pharmacy. He is the associate director also of our Drug Discovery and Development Core. And we also are welcoming Dr. James Polli, Professor of Pharmaceutical Sciences, Professor, and Ralph F. Shangraw, Noxell Endowed Chair in Industrial Pharmacy and Pharmaceutics, University of Maryland School of Pharmacy. Also, he is the director of our Drug Discovery Development Core. Please welcome us or join us in welcoming Dr. Shapiro and Dr. Polli in today's presentation. Dr. Shapiro?
Thank you, Wanda, and good afternoon, everybody. I'd like to take a few moments today to talk a little bit about the opportunities available to UMB scientists in the Drug Discovery and Development Core. I'm going to start at the beginning, and then at the last half, Dr. Polli will talk about some specific examples of translational research activities on campus. Oops, I was trying to advance here. There we go. So just a little bit of what I want to tell you about today. I'm going to give a brief overview of the Drug Discovery and Development, or D3 core services, and how that ties in with colleagues over at Johns Hopkins University. I'll give you some specific examples of services offered with each of the cores here at the UMB campus. And then I'll talk briefly about how you can initiate a project with these core services. Dr. Polli will follow up with some examples of specific projects at the GCRC. So the Drug Discovery and Development Core, or the D3 program, was a collaboration between the University of Maryland and Johns Hopkins as part of the CTSA that supports clinical and translational research. The real goals of this is to provide a state-of-the-art support, both from concept to consulting and technical expertise for both translational scientists within the University of Maryland campus, as well as Johns Hopkins. And hopefully this program will provide the infrastructure and expertise so that scientists can navigate this very complex drug discovery and development process. So the core services that I'm going to focus on today are listed here, and these are the ones on the UMB campus. So I'll talk about the computer-aided drug design, mass spectrometry, the drug formulation lab, and the clinical pharmacology and pharmacometrics. And just as a bottom line, each of these core services provide scientists with 25 hours of free research-related consultation, in addition to that, technical expertise. And I'll get into some details on that later. This is just one part of the D3 program, though. And the D3 cores really encompass both facilities at the UMB and the Johns Hopkins campus. And the basic goal is to provide the expertise and technical expertise from all steps of the drug discovery and development process, from early drug discovery to the development of pharmaceutical products and regulatory aspects, to applications in the clinic. And all of these items here listed are covered by either the Johns Hopkins campus or the UMB campus, as indicated by the different colored boxes. And CROs will be available in case things like toxicology aspects are needed. So I'm going to just talk a little bit about each of these core services on the UMB campus. And the first one is the Computer-Aided Drug Design Center. Alex McCrell is the core leader in this service. He provides computational and database information with the ultimate goal of discovering novel chemical entities that can be developed into novel therapeutics. And just how this, just a real brief how this works is this: if you have a protein target and the computational aspect can look at that protein and identify regions that could be druggable. And then specific regions of interest, highlighted here in the box, can be used to design molecules that fit in specific regions. And that's just shown here in this structure here. And the cages, the different colored cages that are shown here, are potential chemical entities that can be added onto this molecule to improve its binding, to improve its drug-like properties. And some aspects of the and projects that are going on at the Computer-Aided Drug Design Center are the development of antiviral agents. So in collaboration with Matt Freeman in the School of Medicine, he is developing inhibitors of RNA polymerase enzymes that are involved in viral replication. In collaboration with Dr. Jeff Hasday and myself, we are developing anti-inflammatory agents. And this is a project that has gone from a concept of developing function-selective kinase inhibitors to treat inflammation associated with acute lung injury, to licensing of this project. And hopefully the first in-human trials will be at the end of the year. And so this was a project that was really benefited from the ICTR resources. And then anti-cancer agents with Fengxian Chu in the School of Pharmacy is developing novel inhibitors of this protein called BCL6 to treat various cancers. Moving on to the next core service, the Mass Spectrometry Center. Dr. Maureen Kane is the director of that core service. And they can, in addition to providing the free consultation, they can provide subsidized rates for sample analysis and instrument time. And the important part of this is that it really involves communication with the core leader, because the instrument time and the scope of your project, it can vary. And so the subsidies will really depend upon how many samples and what you really want to analyze. But some of the things that can be done here at the center are differential protein expression profiling. And you can get these heat map type of information, large data sets of changes with protein expression under various perturbations. You can look at post-translational modifications. And importantly, you can analyze this data using bioinformatics and pathway analysis software to generate these connectivity maps in your system. And some specific projects that have been supported through the ICTR on campus are listed here. So in collaboration with Dr. Muad'Din in the School of Medicine, using animal heart transplant models to develop proteomic profiles. This generated preliminary data for a U19 application that was funded, with Marta Lipinski looking at proteins in specific organelles, like the lysosome or the mitochondria. This also generated preliminary data for a UMB-UMCP Challenge Award that was funded, and an R01 application that is being reviewed. And then the last example is in collaboration with Mandy Oglesby in the School of Pharmacy, looking at metal binding proteins in the context of Pseudomonas and infection. And these projects have led to a manuscript and also data for a competing RO1. Moving on to the next core, the Drug Formulation Laboratory. Dr. Steve Hogue is the director. He leads a team that will allow you to prepare and develop formulations of GMP products and matching placebos for both preclinical and clinical studies. These preparations adhere to all FDA standards, and they can provide services that are in all steps of the development process, from formulation to manufacturing, packaging, and shipping. And some specific examples of that and the collaborations that the ICTR has supported are the development of some anti-cancer agents in collaboration with Drs. Kundu and Fulton in the School of Medicine. The formulation lab has taken taro root extracts and separated these extracts using chromatography and looking at these extracts in terms of their ability to promote survival in an animal model of breast cancer. And that's just what's shown here in this survival curve. Another project with Dr. David Gorlick in the School of Medicine is developing therapies for treating alcoholism, alcohol addiction, and so preparing drugs that have biomarkers like riboflavin to monitor patient compliance. And the last core service that I'm going to tell you about is the Clinical Pharmacology and Pharmacometrics core. Dr. Joga Goburu is the core director there. And he basically focuses on modeling of pharmacokinetic and pharmacodynamic parameters for experimental and clinical trial drug design and development. And so he can generate, his group can generate models of drug dosage over time that achieve certain required goals as far as drug concentrations. And these models then can be applied to experimental parameters, and then experimental data can show that the models do support, again, achieving a certain concentration of drug in the patient. And some examples of the Clinical Pharmacology and Pharmacometrics core service are indicated here. So with Dr. Eric Strauss, he's looking at drugs that regulate blood coagulation in the context of cardiac surgery, looking at, and with colleagues at the Johns Hopkins University, looking at antibiotics and the treatment of different types of infections, and developing models on monitoring where does the drug end up, and looking at, for example, various compartments within the body, both the region of interest and various other compartments, the metabolism of these drugs, and developing models that can be used to monitor drug doses, drug concentrations, and efficacy. So that's just a little bit about what these core services can provide. What I want to do now is give you a little bit of insight on how to take advantage of these core services. And it's a very straightforward process where you go to the website and you can log in and you can ask for what you would like to do. The 4 cores are listed here, and what I really want to emphasize the importance between the communication between the client, the researcher, the service core director, and then the ICTR leadership. And so this is an important aspect to make sure that you can get what you need to done in your research program. So if you go to the website, if you go to the ICTR website, you can navigate this website and apply and make a request through REDCap. And it's a very straightforward request. And it's not a complicated application. This will be reviewed by the ICTR team. They will send this to the core director, who will then contact the client and discuss the scope of work, other aspects, what they want to, what are their goals, what do they want to do, and how we can achieve these goals. The important aspect of this is that this gets entered into our database through the REDCap, and so that the ICTR administrative team can monitor the requests, can request for updates, progress reports, and then also record milestones and achievements of these projects. The client works closely with the core leaders to achieve these goals. They respond to our requests, which are very easy to submit, and also finishes their final reporting. These services are also available to Johns Hopkins faculty. They generally can just contact the core leaders directly, and then they will provide them access to the application process through REDCap. So it's a pretty straightforward process. It's not onerous. It's a very straightforward way to monitor these applications and to achieve the goals of the scientists. This is just a screenshot of the ICTR Drug Discovery and Core Services page. And so you can, if you just log into the ICTR website, which is available on the UMB webpage, you can get information on each of the core services. You can click onto those, and that'll take you to the REDCap. The REDCap can also be accessed through this page. And then that will provide you with the instructions that you need to make an application for one of these core services and take advantage of these services. So that, I think, covers pretty much all I wanted to talk about. I'm now going to turn over the program to Dr. Jim Polli. He's going to tell us a little bit about research projects at the GCRC. And I also have Dr. Audra Stinchcomb, who some of the projects that he'll talk about will be indicated here.
Okay. All right. Thanks, Paul. Hopefully you can hear me. So my name is James Polli. I'm a colleague of Paul. And what we'll do for the next couple slides is kind of just amplify what Paul described in terms of the drug discovery core, and talk about, some of the translational things that can be done on campus, in particular using the GCRC, which is on the 10th floor of the hospital, as you may know. And so the two people, I got some slides from Audra Stinchcomb, she's like me, a formulation person. Paul mentioned the formulation lab that Dr. Hoig, the GMP lab that Dr. Hoig heads up. So we all work in this space. Now that I just thought about it, all the examples I have to show you don't involve using the GMP, but we've certainly made products here on campus. In fact, one project I have ongoing deals with a very poorly soluble compound, less soluble than marble. So think the Venus de Milo, the solubility of this drug substance is even less than that. And so we're testing to see whether some formulation approaches actually can enhance bioavailability. So we often think when we do pharmacokinetic studies, we think about pharmacokinetic profiles, and we kind of compare things. So one thing we often think about, particularly with things that are funded by the FDA, we have a little niche there. So the bioequivalence standard has to do with whether two profiles are very similar or not. Okay, next slide. And here are just some examples from Dr. Stinchcomb, just 3 examples. She conducts many studies at the GCRC. I think she has 3 studies ongoing at this very moment. I'll just maybe read to you the scope of some of them: the absolute bioavailability, pharmacokinetic, and residual drug analysis of duragesic transdermal system and generic. So again, Dr. Stinchcomb happens to be a person interested in topical skin delivery. Determination of serum oxybutynin levels after using oxybutynin transdermal delivery system and gel, different type of formulation with and without heat. So looking at some consumer issues about whether one applies heat, whether that has any pharmacokinetic effect on transdermal drug delivery. And then also using one involving sunscreens. People can use sunscreens, they put them all over their skin. And as it turns out, she's shown that there's a lot of sunscreen absorption after applying to the skin. Next slide. Okay, so kind of putting our clinical operations caps on, so what is it that the GCRC staff do for some of these pharmacokinetic studies? They administer formulations, they take blood samples and they process them, they assess skin irritation, they take vital signs. In these skin studies, looking at patch adherence scoring, apply heat. So, these are very complicated studies actually that are done at the GCRC. Okay, next slide. Again, kind of from a more operational standpoint, what do some of these studies look like involving fentanyl, anti-pain drug, as you may know. A typical PK study is on the order of a few dozen. Transdermal patches are applied and compared against an IV formulation to do some sort of pharmacokinetic calculations about bioavailability, usually multiple sessions over many days. In this case, there's an opioid dependence challenge because fentanyl is an opioid. Always serial blood sampling, vital signs, and then other things that are related to patch studies in particular. Okay, next slide. In a sunscreen study, again, it's a pharmacokinetic study. So, in this case, just a dozen volunteers, sunscreens applied multiple times a day for many hours. Subjects come in for multiple visits. And as you can maybe see from some of these images here, I don't have a pointer, but towards the bottom, heat is applied in one photograph, and then humidity is applied in another, heat and humidity in another photograph. Okay, next slide. And then as a pharmacokineticist, we love to see this sort of thing. These are some pharmacokinetic profiles where, in the case of nicotine, fentanyl, and Duragesic, I forget what drug that is, but so maybe just looking at this one here, Nicoderm CQ, a particular product, looking at the effect of, I think, when is heat applied, or so-called early or late. And so this is very fun to look at this sort of thing. Okay, next slide. So those are some studies from Dr. Stinchcombe, just another study from myself. I think all of those involved healthy volunteers. Here's a study we looked at in patients, in particular epilepsy patients. So physicians kind of wondered a lot about the whether bioequivalence that was demonstrated in healthy volunteers, which is sort of the standard, whether that applied to actually patients. So we did that study. And here are just the pharmacokinetic profiles of brand and generic when done in patients. And you can see the pharmacokinetic profiles are very, very similar in these patients who are taking their epilepsy medicine every day, or every 12 hours, in fact. Okay, next slide. And then in pharmacokinetics, one term you hear quite a bit about is variability. So in this particular study, it was a replicate design. So each patient took brand and generic twice, actually. And indeed, there was some variation in the blue. You can see that there was variation between the two occasions when the generic was taken in the same individual. But that variability was basically the same as if that same person was taking it in brand. So again, we didn't really see any difference in means or variances between brand and generic. Okay, next slide. And then as a follow-up to that study, which investigated one particular drug, the most sort of problematic drug in epilepsy, we started to just look at all sorts of different anti-epileptic drugs. And we actually did this study to focus on patients. So some patients, about 40% of patients really complain about generics. So we kind of did a study that was highly developed with them in mind, regardless of whatever anti-epileptic drug they took. So we did a study very similar to the previous study involving lamotrigine, but where different drugs were looked at, epileptic drugs, where again, it was their patient's own anti-epileptic drug regimen. Next slide. And then, so this was a relatively small study, just 16 subjects, but the vast majority had a prior switch problem, usually with the study drug. And usually it was a brand generic type of switch problem. But the most common type of switch actually is actually between generics. If a patient goes to a pharmacy and it's generically available, it's very common for them to get a different generic every month, depending upon price. Okay, next slide. Just a little more details about what I think is a very interesting study. Obviously, this draws on the patients that come to campus for treatment. but the study drug was the most problematic anti-epileptic drug in virtually all the subjects that were studied. We did sort of relate issues about disliking generic to some other things, such as, for example, in the last bullet point there, how many medicines do they actually take? As it turned out, people who take more medicine complain more about generics. Okay, next slide. And then here's kind of a summary of the findings, just looking at this last column in terms of pharmacokinetic assessment. Is really the same in all the subjects. It really wasn't a bioequivalent study where everyone takes the same drug. In this case, we had a very interesting pool of patients, and then we challenged them with their own medicine of interest, brand and generic, or in some cases, generic to generic. And as it turned out, all the PK profiles were the same. Okay, next slide. And then I think the last example we'll show deals with IV iron, which is used to treat anemia. And so there's many different types of iron products. The one brand product deals with is denoted to be Ferlicit. In particular, that's a sodium ferric gluconate, and there is a generic of that. And we wanted to go back and do a phase 4 safety study, basically. And the thing that's kind of interesting about sodium ferric gluconate or iron IV products in general is they're so-called complex. That's actually a term that's used. And in this lower left corner, you can kind of see this this nanoparticle. See this 2 to 8 nanometers. So just the composition is very complicated. It's not really a solution at all. It's not a simple solution at all. Next slide. And a lot of, Dr. Shapiro described that there's a lot of laboratory capabilities. So a couple of years of effort went into this. This is in Dr. Sarah Michel's lab, as well as Dr. Maureen Kane, looking at all these in vitro physicochemical properties. And in some cases, they were the same. In some cases, they were different, particularly in terms of like size of the nanoparticle and how fast the nanoparticles disintegrate. Okay, next slide. And on top of that, one thing that's really complicated about these iron products is it's not just iron, there's different types of iron. There's so-called transferrin bound iron, non-transferrin bound iron, and people really didn't have a good understanding of what non-transferrin bound iron was, which was a big part of this pharmacokinetic study in general. So Dr. Kane and Dr. Michel spent probably about 2 years developing an assay for all these different iron species. Okay, next slide. And so from all of that, a phase 4 safety study was conducted, but it was a pharmacokinetic type of study. Okay, next slide. And then, so in terms of pharmacokinetics, we looked at all sorts of different iron species. It's kind of abbreviated here, sorry about that. But the first one was sort of the key thing we really focused on, this so-called drug-bound iron. So actually able to measure the drug, these particles in plasma. And AUC has to do with kind of how much was there over the entire time frame, this area under the curve. And we see that in red here, this was not too far from 1. So we concluded that they were, that the generic was not inferior. Okay, next slide. And then here's some of the pharmacokinetics of these different iron species, total iron, drug-bound iron, transferrin-bound iron, and so-called label iron. Iron's very toxic, so this was a concern. Okay, next slide. And then looking at our primary output in terms of so-called drug-bound iron, this is pharmacokinetic profile of drug-bound iron. And after an infusion, and then we see it goes down within a few hours, and it was really the same, indistinguishable between brand and generic. Okay, next slide. So we would conclude from our primary outcome that they were kind of the same. We did see some differences in adverse events. The nurses, of course, monitor the adverse events. And here's a plot of subject number versus time of start of adverse event. And this is for generic, and they were all just sort of local adverse events. There were 3 of them. Okay, next slide. And then there's a lot more data points on here. This isn't, and you can see going across left to right that some of this was in the same subject, of course. And there was a few subjects that kind of dominated this plot. But there was, lower back pain, hemodynamic systemic side effects, non-hemodynamic effects. So there, we didn't really do calculations, statistics, but seems as if there was a bit more side effects in the brand, actually, particularly in women. But we didn't really look at that closely. It wasn't designed for that. I think that's probably the last slide. Thank you very much.
Hello, everyone. If you have questions, please add those to the group chat, and we're happy to bring those to our presenters today. If not, we could give a few more minutes if someone has some thoughts. If possible, you can use the chat feature. I think you can, if you click on that, you can use the raise hand feature if you would like to speak. If no one has any questions, the one thing I just want to highlight again is that the GCRC is the General Clinical Research Center. It is a clinical research resource on our campus that provides nursing services, blood drawing, blood processing. There's space to conduct research. That is a resource that is eligible for the ICTR voucher program. And I did add that link to the GCRC into the chat, so you can look at that there. And as I'm speaking, I do see that some folks are having some questions. So let me just take a second to bring those to our presenters' attention. So Dr. Polli and Dr. Shapiro, one of the questions is coming from Dr. Susan Dorsey. She is asking, are these services for high throughput screening available in this core? Vana, if you could unmute Dr. Polli.
Yeah, I think the question was for Dr. Shapiro. So I think I can answer the question though, while you unmute Dr. Shapiro too. Yeah, there are. Yep, go ahead, Paul.
Okay, thank you. I'm sorry, I was trying to scream. The mute wouldn't work. Yeah, Susan, there is, we do have, it's not part of the ICTR, but there is a high throughput screening service that we have here that has a library of FDA, all FDA approved drugs and related biological molecules. So I'd be happy to talk to you about that if you wanted to set up something. And we do have a high, I would call it, you can't screen hundreds of thousands of compounds a week, but you can do 1,000.
Another question we have is from Dr. John Hamlin. This question is for Dr. Polli, and he asked, when you see differences between the PK for brand versus generic, do you ever go back to determine whether the active agent is the same in each formulation?
Yeah, hi John, how are you? Well, we do, as Dr. Shapiro mentioned, we do have a GMP facility, and we probably do about one study every 2 years in the GCRC where we take formulations that we've made and test them in the clinic. So we're certainly aware that formulation can make a difference. We actually have never seen where there was actually differences in marketed brand versus generic products. We just haven't seen that. I did show you that one example of IV iron, and we have primary aims and secondary aims. That study was just not powered. So I think in our protocol we said we wouldn't even do statistics, so that's why I'm not showing you any statistics. But obviously a lot of these brand generic studies are actually funded by the FDA, and we certainly tell them what we observe. But for IV and iron, for example, we haven't gone back and done any more testing. Although we have some ideas though for what we should do.
We have another question, Dr. Polli, from Dr. Fulton, Dr. Amy Fulton. She's asking, do you have experience in PK studies for high MW proteins?
I personally don't, I think we can do them, though. I think their design tends to be very similar regardless of the molecular weight. And as Dr. Shapiro explained, there's a mass spectrometry center, Maureen Kane, who I routinely work with, is the executive director of that. And I forget how many mass spectrometers does she have, Paul? But yep, there's a lot of people that can analyze proteins.
That could be a project that you could discuss more, Amy.
Are there any additional questions or thoughts from our participants? So Dr. Dorsey has an additional question. She asked, does the core have experience targeting RNA for small molecules versus proteins? Can CAD type assays be done?
So, Alex has done nucleic acids. I would talk to him about that. He's done other biomolecules besides proteins. The structural organization is a little different, so you'll have to talk to him about how that might work. But yeah, he certainly has done nucleic acid modeling.
If we don't have any additional questions, I just want to remind the participants that I have included a few links in the chat area. One is to the ICTR website where the enrichment seminars will be posted. This session is recorded, and this recording and the PowerPoints will be posted. We also have the link to the ICTR survey in the chat, and the QR code is on this final PowerPoint slide. I also included the ICTR website link to investigator resources that highlights not only this core, the Drug Discovery Development core, but other cores such as biostatistics, informatics, our community engagement core, our studio sessions as well. So please take some time to review those resources that are available to the researchers on the campus. And also, I've included a link to the General Clinical Research Center as a clinical research resource, and you can find out more information about that resource and maybe even how to apply for voucher support if you're interested in using the GCRC for your research. If anyone else, if there are no other questions, oh, I see that Richard has a question. Can the DDD do formulations for non-human, I'm not sure if this is primate or primate. Okay, primate studies.
Yeah, I don't know that that's been done. Oh, I don't know that primates have been done before, but certainly humans have been done before, as well as non-humans and smaller. So just the thing that was actually going through my mind and reading the question was, primates are smaller than humans. So when it comes to designing dosage forms, you need to sometimes accommodate size and of course dose, but I would say yes.
Great. Well, thank you everyone for some really great questions. If we have no further questions.
Okay.
Thank you everyone for attending today's session. We look forward to inviting you again for another enrichment seminar series in the coming months. We'll be sure to notify everyone when that is posted. And again, thank you, and everyone have a great rest of the day and upcoming weekend. Thank you so much. And thank you, Dr. Polli and Dr. Shapiro, for taking time out to bring us this presentation. And everyone have a great rest of the week. Thank you.
Thank you.
Thanks, everyone.